Today we're going to be discussing our lab on cell viability, and we use an indicator reagent called MTT. So these are MTT assays that we'll be doing today. I'm going to talk to you about doing assays and about the whole concept of cell viability and how it follows nicely from what we've been working on with cell counting and doing growth curves. So in general, when you think of the type of data that you can generate from cells, you know, there's the observations. Right? So and we like to encourage you guys to make your observations, to look at cells. You know, we've got these microscopes in the lab. We're always taking plates and flasks out of the incubator, putting them on the microscope stage, focusing in on them, looking at them. We look at them constantly, sometimes just for routine purposes, like to see if they're contaminated, if they're healthy, if they've been dividing, are they confluent on the plate, are they subconfluent? You can get a quick idea just by looking at the cells in the microscope. So that's kind of why, you know, one of the default positions in a lab that works with cells is, well, look at the cells, see what's going on. I mean, you're not going to want to use a plate of cells even for some other biochemical or molecular analysis if the cells don't look good. If they're contaminated, that's not going to be any good. If there's not enough cells on the plate, you're not going to be able to derive protein or nucleic acid from that, right? So you need to always kind of look at the state of the cells. So those are observations. And it's kind of a caricature of the way biologists are, right? They're always looking at microscopes. You know, people, that's the way people think. It's like, oh, we're always, you know, just looking at cells. Well, how much looking can you do to really, you know, what are you going to understand by seeing a bunch of cells? But there are a bunch of things, and then we'll be getting into some of the enhanced digital imaging of cells actually in the next class. So cell imaging and the use of markers, immunohistochemistry. You know, those are great techniques, and the digital imaging age has really made observations a big deal. So that's all fine, but what we're going to talk about today are ways to make measurements. So these are pretty powerful too. I mean, observations take you, you know, so far, but if you can measure something, you know, if you can get some data, collect data on some aspect of the cells, then the measurements are very valuable. And, you know, typically if you're looking at graphs, you know, bar graphs, line graphs, there's graphical representations of data. Those data were collected by making measurements, right? So what we're doing today is looking at one way that we can make measurements. So what can you do with this data that's based on measurements? Well, you know, one of the things is just overall viability. Well, how is that different than observations? I mean, you can look at the cells and see if they're viable or not, right? But not completely. I mean, you see sort of these round things, and you think, oh, that's a cell. Is it a living cell? You know, we haven't gotten into viability staining yet. There's a reagent, several reagents, but trypan blue is the typical one used to look at cell viability. But you know, even the counting thing that we're doing electronic counting. So we trypsinize the cells in the dish, and we make a slurry out of that, and we take our cell suspension and we count that in the cell counter. You guys have done a lot of that week after week. We've been counting cells and doing the growth curves and that. But do you really know that the cells are viable? Think about that. I mean, the electronic counter, how does it count again? It was one of the questions on the quest. How is the electronic counter that we're using, how does it detect a cell? You guys know this, right? It detects a cell. You know, there's an aperture, which is a hole basically. So this is a small pinhole in the side of the tube here, that pinhole, and then this whole thing is sitting inside of your vial, and it has your test sample in there. Right. So then, as your test sample is drawn up into the pinhole of this tube, it comes down the probe, you could say. The hole we could call the aperture, the tube, we could call that the probe. Then it's got a vacuum, and it's vacuuming up some of this cell suspension, and it's going through this tiny pinhole. And as each cell goes through, there's an electrical resistance, because cells are not conductors, they're insulators, because they have a lipid bilayer, that kind of thing. So there'll be a momentary deflection in the electric field. So the other thing that's in this vessel is a metal plate. You should take a careful look at that today. There's a metal plate that comes down. It's a silvery plate that's hanging out inside of your vial as well. And so that's, that and another, inside of here, there's another electrical connection connecting this fluid to electricity. So there's a field that goes across those two plates, right? You're used to this basic understanding of electricity. When you plug in anything at home, it's got two wires, right? One is the hot one, and one is the ground. The current is flowing. So there's a flow of current, and the cell is an insulator. When it goes through the pinhole, you see that deflection. Okay, but that's a total cell count, but not a count of viable cells. Typically, our cultures are almost entirely viable cells, right? So when we're growing them and we're nursing them along, using nutrient medium and all that stuff, they're mostly viable cells. So for the cell counting that we're doing, that's fine, because 99+% of them are viable cells. But you can imagine other scenarios where there would be lots of dead cells, and you might be interested in killing some cells. You're interested in nurturing and growing cells, but killing cells. Why would you want to kill cells? Give me an example of an evil cell that needs to be killed. Cancer, yeah. There you go. So cells that are out of control, it'd be nice to prune those back and actually get rid of them completely if possible. So, so that it brings up this whole question of toxicity, right? So you have viability, but you also have toxicity. So in certain scenarios, you might want there to be toxicity. You want to get rid of the cancer cell. On the other hand, there could be environmental reasons why you want to screen for toxicity. You don't necessarily want toxicity. So I'm drinking some orange juice here. I'm hoping that there's no toxic components in the orange juice. We could test that. We could incubate this with some cells. See what it does. Anyway, so environmental toxicity, food toxicity, there's a lot of reasons to screen for toxicity. So when I say screen, that introduces the idea of drug screening. So drugs are typically small molecules. Right, they're small organic molecules. What kind of chemistry are you guys taking? You're taking organic. Okay. So life is is an organic chemistry problem, or it's a mixture of organic molecules, and that's carbon-based molecules, and that's really what a lot of life is based on. But so drug screening, we make small compounds, and they're often organic compounds because we're organic ourselves. And so we come up with drugs that will control things within our bodies and also control infections, control other things, bacteria, things trying to live inside of us. So drug screening, a type of drug screening, we already mentioned this. You know, cancer drugs being one example where you're interested in toxicity, but you're using it in a useful way. Another example is ingredient screening. Right? So that's actually a pretty big deal when you start to look at this. It's like, wow, this is a lot of stuff. So what we're talking about is all the drugs that you guys, are exposed to or can take if you get sick. The doctor's gonna, give you a prescription for certain drugs. How were those drugs found? They were, they were synthesized and they were screened using a variety of different screens, adding the drugs sometimes to cell cultures. It's not the only way to screen drugs. You could do experiments with animals. You could kill thousands of animals trying to figure out which drugs are the best, and in fact that's been done quite a bit. But but cell screening seems like the place to start because you can screen a lot faster and a lot more and understand some different things. So you can understand cell toxicity, you can understand positive and negative effects, but for cancer drugs you might want specifically to have the toxicity. But ingredient screening, it's like, okay, so we're human and we are interacting in the world, and a lot of our environment we control. We're sitting in a room now that was entirely made by human hands, right? The floor, the furniture, the whole thing, right? So we create these environments for ourselves. But, the things that we're creating are coming in contact with us. They might be things that are actually consumed, or they might be things that you touch, or you smear on yourself, or you take a shower, and you use some soap, and you use some shampoo, and you use some toothpaste, and you're using these treatments or consumer products, that kind of thing you get into, and they all have ingredients. If you read the bottle, it says it's got these different things in it. Well, how did they figure out to put this stuff in there? How do they know that that's good for hair? How do they know it's okay for your scalp to be exposed to certain concentration of sodium lauryl sulfate, and for how long and all that? So I mean that's the kind of thing that what we're doing today has implications for. So if you were going to get an introductory job out of college working in a lab, there's a huge possibility that you'll be, if you're doing some kind of science-related lab and it's biological, that you'd be doing some sort of measurement, screening something. And the reason is these are products. Products have revenue, right? So the pharmaceutical industry is huge, and it has a big presence in the Bay Area. I worked at Genentech. Genentech was bought out by Hoffmann La Roche. It's a huge pharmaceutical company. Chiron over in the East Bay is another biotech company that was also sold to Novartis, another Swiss pharmaceutical company. So the pharmaceutical companies are huge. These guys have so much money. They can make acquisitions for tens of billions, no problem. So that's just drugs, and then ingredient screening. I mentioned all these products, soap and shampoos and toothpaste and all that stuff. I actually did some work with Procter & Gamble at one point. They're interested in some of the cell-based assays that we were working on, and they're interested in screening compounds. And a big conglomerate like that, besides making the soaps and all that other stuff, they also make cosmetics. So Max Factor and CoverGirl, and those are Procter & Gamble products. So it just gives you an example. It's like, wow, so this is a big chunk of industry and a big chunk of pharmaceuticals or a big chunk of medicine, and so here we're doing some measurements on cells, but that's what you do in order to develop products like this, and those are the employers. And if you're in medicine, you're going to be administering drugs, and those drugs were screened, and they were screened, at least partially with cell-based assays. So the old thing used to be test tubes. You guys work with test tubes, maybe in chemistry? You know, the glass test tubes, and you're doing a reaction and you're holding the test tube and you're heating it or whatever you're doing. Well, that's nice, but you know, it's kind of cumbersome. You know, you've got a couple of mils of solution and you're adding some stuff to it and you're swirling this test tube around. That's kind of inefficient, right? I mean, you can, you know, we live in a world where everything has been, you know, made using a microchip, right? I mean, the electronics have been shrunk down to a tiny little etched, you know, circuit on a silicone wafer hybrid. So instead of a test tube, you know, biology is trying to move in that direction too, into the micro stuff. So we use these plates, and these are multi-well plates, and they've got, so these little indentations we're calling wells. So a multi-well plate, like this one, is a 96 well plate that we're going to be using today, which gets on the verge of sort of high throughput type plates. There's other plates out there, 384 little wells on a piece of plastic. So these multi-well plates are, you know, kind of a modern adaptation. So rather than having a glass test tube and your data equals, you know, 1. You know, that this experiment that you did is like equal to that 1 well. And then there's 95 other wells you can do at the same time. And then how do you handle this? I mean, you can, we're pipetting microvolumes, so we use the, you know, these micro pipettes, Pipette Man being one brand. And they're multi-channel pipetters. So rather than pipetting up and down solution with just one channel, you could, you know, have 8 at the same time, or you could have 12 at the same time. Actually, this plate is 8 down and 12 across, so it depends on whether you want to go, you know, down this way or you want to be going sideways. If you're going sideways, you can get away with the 8 channel multi-pipetter. So those are tools. You know, those are tools that allow you to do much more. I mean, basically, you know, you're doing almost 100 times more than you were doing with the one test tube by using multi-well plates. So that's kind of cool. But you know, and then these plates have machines that they're coupled with. You know, the measurements are made using machinery that is designed to read these plates, either 96 well format or 384 formats. And so by having machines that are electronically detecting something, you know, you're getting the results much faster, and it's more quantitative. So what are those machines? Those are plate readers. So these multi-well plates are read in plate meters. And you know, typically what they're measuring is some sort of light. Light is nice. It's very useful stuff. Because it doesn't interfere with the sample, right? If you shine some light on something, you're not destroying the sample. So visible light. Ultraviolet light.